Good morning. Our topic for today is the continuation of your past lessons, that is on GMO and nanotechnology. I have here a very interesting question, and you can see this on the screen. Is our future written in our genes? So you will answer this after we are finished with our lesson. So this will bring us to our topic for today, that is on gene therapy. First, let us understand or give meaning to genes. So what are genes? Genes are segments. So these are segments here. So these are segments in the DNA that supply the body with instructions, directing the cell to produce proteins that perform a vast array of functions in the body. So those are the genes. So genes are important because the genes are for the various structure and functions of our body. However, our genes are not always in good condition. So there is also conditions where there are defective genes in our body. And as a result, this could be failure of protein production. A defective gene could also be resulting to the production of a non-functional protein, as well as overproduction of protein. So all of this will lead to some sort of a disease or disorder. So what is now the cure? Not really a cure, but that is how to overcome these defective genes. So we now have gene therapy. So what is gene therapy? Gene therapy is an experimental treatment that will involve insertion of a gene to correct the effects of a defective gene. So this kind of treatment is now to correct whatever genes are not functioning well in our bodies. So the researchers are now into several approaches to gene therapy. So one is they replace a mutated gene that causes disease with a healthy copy of the gene, or they could inactivate a mutated gene that is not functioning well, or there is now the introduction of a new gene into the body in order to fight the disease. So we now move on to the types of gene therapy. So there are actually two types of gene therapy. One is a somatic gene therapy, and second is the germline gene therapy. Now, what is the difference between these two? Somatic gene therapy, from the word somatic, so this would mean body cells. So transfer of a gene to any cell of the body. Okay, and the effect of this is not passed on to the patient's children. Okay, because these are body cells or our somatic cells. On the other hand, there is also the germline gene therapy, and this is not the transfer of a gene to the cells that produce sex gametes. So these are eggs or the sperm. The effect of this is to be passed on to the patient's children. Okay, so these are the two types of gene therapy. Now, how does it work? So how does gene therapy work? So we have here the various techniques. So one technique is known as the gene augmentation therapy. And in this kind of therapy, we have here in the diagram a cell that is not functioning, or with a gene that is not functioning, or is a defective gene. Okay, and a functioning gene is now inserted into the cell, so that the overall result will now be the cell with a functioning or this is now a cell that functions normally. So that is now brought about by the insertion of a functional gene. That is what we call as gene augmentation therapy. Another kind or an example for this is cystic fibrosis. Okay, cystic fibrosis is actually a fatal disease, and this is brought about by a defective gene that produces a non-functioning plasma protein. So the result is the overproduction of mucus that will block the airways or the different ducts in the body. So in gene therapy, there is now the gene that is now inserted into these cells in order that this will now result to functioning plasma protein. Okay, so that is one application of gene therapy in cystic fibrosis. Another kind of therapy is gene inhibition therapy, and that is in this diagram. This is the cell with a faulty gene, insertion of a blocking gene. This blocking gene will now have an effect on a faulty gene. So this will block the effect. It will now impair the faulty gene. So that blocking will now result in the cell that will function normally. Okay. Now, other ways of gene therapy or other techniques will include killing of a specific cell. And there are actually two ways in doing this, where this will involve the suicide gene and then also the markers that are introduced into the cell. Example is in cancer cells. Okay, so in one of the techniques, this is the disease cell. The gene that is now inserted, that is the suicide gene. This suicide gene is now going to release or produce toxic products that will kill the deceased cell. Okay, so it will not anymore affect the individual or the body of the individual, because the faulty gene is now killed, or this is now removed from the body. The second one, as I mentioned earlier on, is with the use of the marker gene. So this marker gene is to be expressed on the cell surface. With the expression of this will now trigger the immune system cells. So our immune system cells will attack this faulty gene and will result to the death of this faulty or these cells. The cells carrying the faulty genes dies in the process. Okay, so these are the various techniques used in gene therapy. Okay, now I have assignment for you, and that is you are going to watch a video that is Boy in a Plastic Bubble. Okay, this is a very classic example of gene therapy. It actually paved way for the progress or the advancement in gene therapy. Okay, so I have this in our Google Classroom. So there is a link, and I would like you to watch this, and then there are questions that are provided for you to answer. Okay, these are our references. Thank you for listening. Have a great and safe day.